Okay, Prof. Me and Kurt and Mandisa, we're gonna vanish. It's just you and Matthew who's gonna be on videos, right? Kurt, have you given me right?
Yeah.
Leslie, will you let us know when we can begin?
Kurt.
Okay.
Kurt.
Yes.
Yes. Can you start? Can you just start, please?
Yes, we can start.
Yeah.
Everything's
Will
live.
you let us know when we can begin?
Kurt.
You can begin.
Kurt. Yeah, Kurt, because I can't see the starting button.
Now the people are coming in already. The people are in
Okay.
already.
So we are live.
Yes, you're live.
Okay.
Good afternoon, everybody, and welcome to another one of the College of Health Sciences talks hosted by our DVC. Today, we're going to look at a topic that is a relatively new frontier. There's not a lot of publications out there. They're coming thick and fast, and that's on the microbiome. People are beginning to look at the microbiome right throughout the human body. I've seen papers on people looking at the ear microbiome and the whole microbiome in the gut, etc. And I became particularly interested in the microbiome when I worked on a paper with one of my students looking at the health of the glycocalyx. And I've realized how the interface between our environment and the glycocalyx in our cells has so much to do with our health. And I've also been in contact with other people internationally who talk about how people with a healthy microbiome got through COVID a lot better than those people who did not have a healthy microbiome. So I'm going to introduce you to our speaker today, who's also been doing some work on the microbiome. And I'm going to tell you exactly what that is. But how I've got to know him over the years is we've been collaborating on genetics work. We've been looking at differences in sex development over the past few years and have started a whole new research project in that. So Matthew Bramble is a geneticist to start, and he has now started looking at microbiomes. So I'm going to read a bit of an introduction on him and then talk to you about how this fits in with the research that we're going to be doing. So Dr. Bramble is currently a Fulbright global scholar based in South Africa and in the Philippines. He's also data sciences faculty member in the Department of Genetic Medicine at the Children's National Medical Center in Washington, D.C., and assistant professor at the George Washington University School of Medicine. Dr. Bramble earned a PhD in human genetics from UCLA, where he focused on genetic and epigenetic mechanisms underlying sex differences in the mammalian brain, as well as genetic influences contributing to differences of sex development, or the DSD projects that we're working on. However, over the last 7 years, he has spent most of his research efforts building a genetic capacity within the Democratic Republic of the Congo to expand user-friendly genetic diagnostic tools, as well as techniques involved in basic molecular or biology genetics. In addition to building genetic capacity and collaborations in the DRC, he has also focused heavily on infectious disease research, such as Ebola. But his most recent research focus revolves around projects dealing with a mechanistic understanding of cassava-induced neurotoxicity, better known as Konzo, which will be discussed in today's seminar. So the microbiome work that Matthew has been doing till now is not just observing gut or oral, etc. microbiome. It's looking at what a specific food type has done to the microbiome in a population. So thank you very much, Matthew, for taking time out to speak to us. And I'm going to hand over to you. We'll have a Q&A session after his talk. So please put questions in the Q&A and we'll get to them after the talk. Thank you very much.
Okay, just let me know if you can't hear me or anything, but thank you for Colleen for inviting me to give the talk, and of course, UKZN, I suppose, for hosting. As Colleen mentioned, I'm going to talk today about a topic that I've probably been working on now for the last 3 or 4 years, and that is really trying to understand how the gut microbiome, or the bacteria in the gut, influences this disease known as Konzo. And to start, so I'm sure many people in the audience haven't heard of Konzo, and I hadn't either really until I had went to the DRC. But Konzo is a neurodegenerative, and it's a neurodegenerative motor disorder, and it's thought to affect the upper motor neurons in the brain. However, the mechanism of this disease still remains largely unknown. As a whole, Konzo means tied legs in Yaka, and that is the local dialect in the region where Konzo is most frequent in the Congo. What happens is it causes a permanent and spastic paraparesis, so essentially a random paralysis of parts of the leg, in addition to cognitive deficits that have really been looked at in the last 3 or 4 years. This disease is quite interesting in that it kind of happens cyclically and during outbreaks. So outbreaks are common in times of drought, war, and famine. And as the presentation goes on, you'll understand why these types of environmental stressors result in outbreaks of Konzo. What's really implicated in the cause of this disease is the consumption of poorly detoxified bitter cassava. And in cassava, there is a cyanogenic glucoside, or a sugar that produces cyanide, essentially, and this is called linamarin. So in addition to consuming linamarin, you also have to have a state of malnourishment, and you have to be low in sulfur-containing amino acids, which are typically derived from eating meat. And unfortunately, a lot of these children that are affected with this disease and in this area of the Congo really have no source of meat protein. So they are on a cassava exclusive diet. And what happens when you are eating this improperly processed cassava, kids are out playing soccer, and then you just wake up one day and you have 3 stages of the disease. You either have a mild form, a moderate form, or a severe form. And this disease is non-progressive, and it's also non-reversible for what we know now. So next, I'm going to show you a video to show you what stage 1, stage 2, and stage 3 Konzo looks like, just so you have a good idea of the disease we're talking about here. So the first video is going to be a child with stage 1 of the disease. And stage 2 here is a bit more moderate, and stage 2 is really classified as requiring some type of device to help walk. And stage 3 is classified as essentially having to be on all fours type of paralysis. So it's a very unusual type of phenotype as a result of eating cassava. And you can imagine if you have villages where, I don't know, 20 or 30% of children at any given time have this disease, you can imagine the stress and morbidity that this puts on the child and the families and the community. Now, it's strange, of course, because cassava is a staple food crop. I believe it's the fourth largest food crop in the world. And cassava is actually native to South America, and the genus species is Maniote esquilante. And this is a resilient shrub, and it is a good food source, of course. There are 2 main varieties that are really grown across the globe, and that's the sweet cassava and the bitter cassava. Sweet cassava is what is grown in most parts of Africa and South America. However, the bitter cassava harbors these really high to lethal levels of lanamarin or cyanogenic compounds. And you may ask, well, who in the world would eat that? But if you have no type of fertilizer, or if you have really poor soil, these compounds actually repel insects, and the plant is able to grow in really harsh soil. So it's really the only source of food that some of these individuals have. So they have no choice but to eat the bitter variety. Now, the removal of these cyanogenic glucosides from cassava is generally straightforward and well understood, of course. So essentially, you start with the cassava plant, and you see me here getting my cassava 101 down, and you just peel off the root, very much like a potato, you would think. And this is where the problem comes in. Really, you soak the root into these holes of water, and the water helps the toxin leach out of the cassava. And you can imagine earlier when I had mentioned drought, if you have a drought, for example, you don't really have the resources to change this water out frequently. So what happens is the toxin builds up in the water, and therefore the cassava becomes ineffectively processed. And of course, if you're in a bind or you're very hungry, you don't really have time to wait. This is a weak kind of process from harvesting to food. So after the soaking, in a normal scenario, you dry it out for a few days. Then it dries and kind of ferments. Down here it kind of gets, this is a little funky, if you ask me. And then after a few days, you prepare it in a traditional dish, which is called fufu. It's quite similar to what you have in South Africa, pap, except South Africa makes theirs with corn, and most of sub-Saharan Africa makes fufu with cassava. A very different texture. And of course, what happens is if you don't process the cassava correctly in the upfront steps, then you have fufu now that contains toxins, the lanamarin, that you were not effectively able to remove from the plant. Now, during the process, so assuming you did not detoxify correctly, what happens is the lanamarin that's in the cassava can be converted to hydrogen cyanide. And it can do so spontaneously or if acted upon by a certain enzyme, the enzyme known as lanamarase or beta-glucosidase, as we will discuss more. And what happens in the gastrointestinal tract, humans inherently do not have beta-glucosidase in their digestive tract, which would be needed to release the cyanide from lanamarin. However, the bacteria that reside within our guts do have that capacity. And once lanamarin is exposed to gut bacteria, they have the ability to utilize the sugar and release hydrogen cyanide. Now, this, of course, is still a problem, but if you have a substantial amount of sulfur amino acids in your diet, say maybe you had a big steak the night before, then you should have enough sulfur amino acids to detoxify this into thiocyanate. But as I mentioned earlier, these children are quite malnourished, so this protein amino acids, or these sulfur amino acids, aren't really present. And the toxicity, and there's an extreme amount of lanamarin coming in. So there's a buildup of this hydrogen cyanide within the guts of these individuals. Now, the question became a few years ago, could the gut microbiome be implicated? And we wondered this because what we see is that despite a very high diet of cassava, only a fraction sometimes of these populations are affected, which is good, of course. But there are instances where 10% of all the children in a village can become crippled within a week of an outbreak of Konzo. And Konzo isn't unique to DRC. It's also observed in Central African Republic, Cameroon, Tanzania, Mozambique, and Angola. And the Democratic Republic of the Congo has the most cases and was the first location to really be documented. But Mozambique, South African neighbor, also has a very high level of Konzo in its rural villages. Now, what's interesting is that this idea that the gut microbiome may modulate cyanide toxicity isn't really new. In fact, it's been around since the 1980s. Of course, I didn't know that at the time, but along the way, after reading, you find these old papers. And in 1980, there was an experiment done looking at how a rat metabolizes this drug known as amygdalin, and then amygdalin is found in the pits of cherries. So of course, you don't eat the pits of certain fruits because they contain chemicals that can kill you. Now, what's interesting is that if you give a rat amygdalin and you measure how much amygdalin comes out in the urine, if you have a mouse that is germ-free, which means that they do not have any gut bacteria, you get 45.5 something. However, if you have a mouse that does have, or a rat that does have gut bacteria, you recover about half of that. So what that tells you is that the gut bacteria is capable of metabolizing these compounds. Whereas if you don't have gut bacteria, you do not nearly metabolize as much of the compound. And again, known in 1980s, most of this work has really been done in the veterinary space due to feeding livestock cassava. And farmers, of course, know that if you feed cassava improperly, or you have very bitter cassava, you can kill your livestock. Another very interesting study in the gut microbiome space in regards to toxicity is the desert wood rat of California. And this rat eats exclusively creosote. And creosote is, you know, kind of like a waterproofing you put on the decks. You know, it's that black, you know, tar smelling kind of compound. And no one could really understand at the time how these rats were able to exclusively eat this chemical. But then they had done a microbiome study, and this is an early microbiome study in 2014, and they had found that this functional ability to metabolize aromatic compounds, as you see down here in the creosote group, are enriched. And creosote is indeed an aromatic compound. So the conclusion at the time was that these rats actually had a gut bacteria that enabled them to eat a poisonous aromatic compound. And because of their gut bacteria, they were able to survive such toxin, whereas other rats and mammals would not. Lastly, what's interesting in this space as well is that researchers have found that certain bacteria harbor high levels of beta-glucosidase, which is the enzyme that you need to release the cyanide from the namarin. Interestingly, this Lactobacillus plantarum is the most effective at lysine linamarin. But what's more interesting is that this lactic acid plantarum, Lactobacillus plantarum, sorry, is the primary fermenter of the cassava before it's prepped for food. So you can think of like a yogurt. We all hear about lactobacillus. So this is kind of like a yogurt type culture in that it ferments, but it also has the ability to release the cyanide from this compound. So you can kind of see where I'm going. If you're ingesting fermented cassava and it has a lot of lactic acid bacteria, and this lactic acid bacteria can release cyanide from the compound, you know, you can kind of see how the gut microbiome could be involved, as I will show you shortly. Unfortunately, you know, not many populations actually rely on a chronic diet of bitter cassava. You know, the regions that I had showed you are really some of the only places that I know of in the world that you can actually query how does cyanide and the microbiome, you know, really work together in a human context. And of course, the central hypothesis to these studies was that susceptibility to Konzo, or cassava neurotoxicity rather, is mediated through variations in the functional gut microbiome. So essentially, we're interested in how does the function of the guts, the bacteria in the gut, modulate this disease. So what we had done, we did a large metagenomic study, and metagenomic will, you know, let us know all the bacteria that are there, all the genes of those bacteria that are known, et cetera. So it's like a, you know, shotgun, they call it. So, you know, when you shoot a shotgun, it goes everywhere. So it's the same kind of concept. We can sequence a little bit of everything in the stool of these individuals. So we did, at the time, it was quite a large study of 180. And we collected 30 unaffected children from Kinshasa, which is the urban capital. We collected 30 from a village known as Masimanimba. And Masimanimba is a rural village that also relies on cassava, but has never had a Konzo outbreak. And then we look at 2 regions within Kahamba. And Kahamba is the area in Congo with the most Konzo. And there are villages of high prevalence, which is denoted here as HPZ. And there are also regions of lower prevalence, and that's LPZ, but all from Kahamba. And we also took unaffected children from these same villages as well. All of this led to a recent publication last year, which is open access. So if you really want to read the details, you can easily download this paper, of course, free of charge. So to get back to it, Kinshasa here is the urban capital of Kinshasa, or of Congo. And if you don't know, African geography, which surprisingly, most people don't, or geography for that matter. Kinshasa, of course, lies right around the equator, Central Africa. Congo is a very large country, as you can see here on the upper left. Masimanimba is about 400 kilometers to the east of Kinshasa, and Kahamba is an additional 400 meters to the south, borders Angola. Now, while we're only talking about 800 kilometers difference here, this is a 3 or 4 day overland journey, given the road conditions and the lack of connectivity in the country. So despite being 300 or 400 kilometers away, you can easily be in a very, very different type of environment and place. So the first thing we really looked at is food diversity. And I don't want to harp too much on this, but essentially we have a scale here that covers how many times a week did you eat X? We ask about maize, rice, oils, vegetables, cassava, etc. And the real take home of this, if you look at Kinshasa over here to the upper right, and you compare that to the high prevalent zone of Kahamba and the low prevalent zone, you can automatically see, without me having to tell you much, that there is more diversity of food being consumed in Kinshasa than in Kahamba. And Masimanimba is somewhere intermediary. It's not as diverse as Kinshasa, but more diverse, say, than these regions over here of Kahamba. And of course, when you do the measurements, they eat cassava 7 days a week, which is also, I don't want to, talk too bad about cassava because it is the staple diet of many, many people. It's just a matter of how you prepare it. And these individuals in Kahamba have a chronic reliance in that, typically it's their only meal is cassava. So when you do microbiome studies, the very first thing you ask of the data is, what does the alpha diversity look like? And alpha diversity is intra-group differences. And a good way to think about this is I always use forest analogies, because these measures are ecological measures from the 1940s and early 50s, and they still hold true today. So when you think of alpha diversity, a rainforest here on the left is considered high in alpha diversity. And it's high in alpha diversity because what's taken into account in this measure are species that are present, the abundance of such species, and how evenly distributed these species are. So you can look at a rainforest and know that there are many, many different types of trees, many, many different types of plants, so on and so forth, which makes it a high alpha diversity type situation. Whereas if you look at the boreal forest, say of Canada, one would say, well, there's a lot of trees, it must be diverse. But if you look at the picture, you really only see one kind of tree. And the scenario is, you have a virus that affects a particular type of tree. If it hits the rainforest, it may kill all of that kind of tree, but there's still plenty of rainforest left. If a similar virus were to hit the boreal forest and it affected these trees, you could wipe out the entire forest because it's not very diverse. And essentially, diversity measures tell us how sensitive to a disturbance is the ecosystem. And this is always the first thing you look at to kind of look at a dysbiosis and overall general health of the microbiome or environment that you're looking at. Because it's much easier just to think of the gut bacteria as a forest and take the human context out for a moment, because that's really all we're doing here is community microbial ecology. So when you look at the very first thing I'd mentioned, alpha diversity, what you see here in panel B is called the Shannon Diversity Index. And this is the very classic kind of measure to look at alpha diversity. And anything above 3 is considered to be diverse, and so on and so forth. But 3+ is considered a diverse, healthy ecosystem. And you can see across the board here, we have Kinshasa, Masimanimba, unaffected, affected from the LPZ, and unaffected and affected from the high prevalence zone. But the moral of that story is that all groups, regardless of disease status, seem to have a diverse and, on paper, healthy microbiome. And when you look at the unique species that we identified after different filtration methods and analyses, between 500 and 750 unique species were able to be identified in the guts of these individuals. And these lists are changing every day. As sequencing builds, more genomes are assembled, and therefore more bacteria can be identified. So if I redid the analysis of the exact same data set, I would find probably 50 or 60 new bacteria because sequences have been produced. Not very important here for these figures, but essentially the take home is that when you look at all of these populations, the overall structure looks to be of a healthy individual. You know, it's not in what's called a dysbiosis kind of state. You know, we don't have time to talk about all these details, so you could read that in the paper, but you can take my word for it that this is what you would expect from someone that is, you know, for all intents and purposes, healthy, even though we know within this data that we have two groups that are certainly in a state of paralysis. So, you know, we're not thinking now that there's any ecological or diversity issues happening with these children. Now, this is where more, you know, a better type of comparison comes in, and this is known as a beta diversity or inter-group differences. And it's a scale, again, from, I believe, 1940 some. Bray-Curtis is the measure. And what you do is you compare, each group in various pairwise comparisons, each species and how they relate to one another. So it takes into account, again, species present, species abundance, and how they relate to each other. But essentially, it gives you a numeric value for how similar or dissimilar the two populations are. So for example, if we do the analysis and I get a 0, then I know we have a completely identical population here. We got two identical twins. Now, if I get 0.5, similarity is high. These are fraternal sisters and brothers, not identical. However, if you get 1, it means that you are completely different. And the joke here, so this is Eric Villain, my long-term PI, and next to him is Quentin Tarantino. And the joke always is, well, are they different? Since they obviously look quite a bit alike. He never finds it funny, but I think I used to have Donald Trump and an orangutan, but I changed it up recently. So that's the idea of the Bray-Curtis scale. And then what happens with Bray-Curtis is it gets converted into a distance kind of matrix. So this is what's called a principal component analysis, or PCOA plot. And essentially each dot is generated after you do this Bray-Curtis analyses. And the closer the dots, the more similar the structure of the bacteria are. And this is a 2D version because this plot actually comes out into a Z-plane, so you can't really determine if they're stacked on top of each other or not. But the moral of the story is if you just look at all those populations from a geographic standpoint, so Kinshasa, Massima Nemba, the low prevalent zone and the high prevalent zone, the unaffected children, statistically speaking, and you have to rely on statistics for some of these measures, is that statistically, these groups are distinguishable and indicating that there is a geographic difference of populations. Now, I could talk about all of these other comparisons, but that would be a bit much. But these circles kind of also help you tell how similar they are to one another. So for example, if you look at panel F, you can see in purple the high prevalent zone, unaffected children in the high prevalent zone, and it's a very small circle. So that tells you that those children are very, very similar to one another. Whereas if you look at the green, which is the Massima Nemba, there's a much larger spread. So it tells us that the variability in the children of Massima Nemba are greater than the variability in the children of Kahamba. So that was an interesting finding. In fact, to find a population that is that tight and similar is very unusual, because you can see here on this panel A, we have blue, which is Kinshasa, which is the most spread. And you would expect that, children from a major city are not eating and living in the same kind of conditions. So you would expect larger differences. And that's exactly what we see here. But collectively, geography is able to separate out these populations. So that was from a statistical kind of old school approach. And then a more reasonable model is to ask the computer to see if the computer can actually distinguish populations blindly, unbiasedly type situation. And without explaining too much, we use what's called a random forest classifier. And again, if you just looked at geography, which are panel A and B, and ask how accurate is the computer at distinguishing what bacteria profile came from what group of people, you actually see here in this AUC, or area under the curve, we can get it around 90 to 95% accurate in distinguishing, did the population come from Kinshasa? Did the gut bacteria come from Kahamba? So on. So that's very interesting. So this kind of adds validity to the old style approach. And we're moving on now, but in panel C, we asked the computer, is there a difference between unaffected children in Kahamba from the high prevalent zone and low prevalent zone? So within Kahamba, are the unaffected children different? And what you actually find with about 70 to 88% accuracy is that they are also different. So the computer can distinguish unaffected children from Kahamba's zones. However, when you ask, are the children with the disease different between the villages? We don't have a very high accuracy here. We get 60 to 65%, which is only 10% above chance at 50. So what it tells us is that the children with Konzo are much more similar in their gut bacteria than children that do not have Konzo. And why that is, we still don't know, but we'll get to that. So as I just showed you from the random forest model, the computer model, which is kind of like figure A here. So this is Bray-Curtis, again, put into a distance matrix. And when you look at unaffected children between high prevalence and low prevalence, again, statistically significantly different, indicating that these two populations are segregating for particular reasons. However, when you look at children with Konzo from low prevalence zone and high prevalence zone, while it does reach statistical significance, certainly not to a strong degree, again, telling us from a statistical standpoint that they are more alike to one another than their unaffected counterparts. Now, what's interesting in panel D and panel E here, so here we compare unaffected children and affected children from the low prevalence zone. And in panel E is the unaffected and affected children from the high prevalence zone. And you can easily look at the p-value here in the bottom right corner, and neither of these are significant. So this tells us that regardless of if you are affected with Konzo or not affected with Konzo, your gut bacteria look almost identical to one another in your respected villages. And at first it was kind of, that's crazy. But when you think about it, it probably should be that way. It doesn't matter if you have Konzo or you don't have Konzo, you're still eating toxic cassava all the day. You're still malnourished. So all the factors that give you Konzo are still present. You perhaps just haven't got it yet. So this kind of led to the conclusion that all children in the Kahemba area are indeed vulnerable to getting Konzo should the conditions be perfect. The real take home of this paper, and this is what I will focus on for the last few minutes of the talk, is that if you remember earlier, I had told you that there were bacteria that fermented cassava, and they were known to have a high ability to release cyanide from Linamarine. These bacteria are called Leuconostat menesterides, Lactococcus lactis, and Lactobacillus plantarum. And all three of these bacteria contain what's called beta-glucosidase, is the gene, and this gene can metabolize Linamarine in the gut. So we ask, do the individuals, so these last four panels here are Kahemba, unaffected and affected. And the first is Kinshasa and Masimanimba. And if you just look at the last four panels together, you can see that there is a higher abundance of all three of these bacteria when compared to Kinshasa or Masimanimba. So that tells us that the children of Kahemba, regardless of disease, have an enrichment of lactic acid bacteria that could potentially make them more vulnerable to releasing cyanide when they ingest Linamarine. So since this was a metagenomic study, we can go beyond just looking at the bacteria, but we can also look at the frequency of the actual genetic sequence of beta-glucosidase, because there are other bacteria also that possess this ability. And again, when you look at the last four panels, these are elevated compared to Kinshasa and Masimanimba. So not only do the children of Kahemba have a enrichment of bacteria that are capable of metabolizing Linamarine into cyanide, they also have an enrichment of the gene that is required to do such. I mentioned earlier that Masimanimba eats the same diet for the most part, but has never seen an outbreak of Konzo. So we wanted to see if there was a difference in rhodinase, the gene rhodinase. Now, rhodinase is the gene that is required to detoxify cyanide into the less toxic form thiocyanate. So in the humans, we have plenty of this in our kidneys and our livers. So should we be exposed to cyanide, we have the genes rhodinase to detoxify this as well, depending on how much we were dosed with. But what's interesting here is that if you look at all four of the children of Kahemba, compared to Masimanimba, we see an increase in bacterial rhodinase in the children of Masimanimba, which is quite interesting, because it means that these children are likely more capable of detoxifying Linamarine than their counterparts in Kahemba, which kind of leads us to this protective or susceptibility scenario. So in the children of Kahamba, we're kind of postulating that there is a scenario where the bacteria makes them more susceptible to developing Konzo. And this happens because, as I showed you earlier, there's an enrichment of bacteria with beta-glucosidase genes. And if you have an enrichment of linamurase, then that means you are going to break down linamarin faster and more efficiently. And if you break down linamarin faster, then you're going to release more hydrogen cyanide in the children's guts that have this bacterial structure. And we know that they have a lower proportion of the gene rhodinase in their bacteria. And effectively, what this tells us is that the structure of their gut bacteria is going to make them more susceptible to developing Konzo because of the bacteria that are present. Whereas in Masimanimba, we see a more protective scenario potentially. I showed you just recently that there is an increase of rhodinase in the guts of these children, which means is there's an increase in rhodinase, then there is likely a possibility to decrease hydrogen cyanide, and you will yield more thiocyanate, which is the less toxic version. And of course, they also have a lower abundance of bacteria with beta-glucosidase. So their ecosystem is more designed to detoxify rather than toxify, whereas the children of Kahamba have a gut bacteria that is more prone to toxify rather than detoxify. So that's essentially the take home. So this is just the very beginning. Of course, the paper is much, much more detailed. But the key finding is that there are unique bacterial populations in certain children in the Congo that likely put them at higher risk for developing Konzo when all of the environmental conditions are met. The high linamarin, low protein, malnourishment, all of those things. So that was the real interest in this study. And of course, we can discuss all the follow-up that's happening. But since how much time do I have? So I'll just go ahead and end here, and then we can take some questions, etc. Yeah. So this is the Eric Verlaine lab here at Children's National. We work closely here with Desiree Chala and Diodene Mumba in the Congo, and Chala is he's really led the way in the Cassava research over the last 20 years or so, and not many people are in that space. So it's a very small community. But with that, I'll leave it there. And if you have any questions or want some more details, just let me know.
Thank you very much, Matthew. That was extremely interesting. We don't have a cassava issue in South Africa, but this is on our doorstep. So I think it's something that we as South Africans should be aware of. And it could get here. And how do we know? How do we not know of other microbiome related illnesses?
Exactly.
So I'm going to open the floor. I saw there was a raised hand very early on in the talk and I didn't take it. Would you like to raise your hand again? Okay, that person hasn't. I'm opening the floor to questions. So please just raise a hand or put your question in the chat. And while people are thinking about how to pose their questions, Matthew, what does this research in the Congo actually mean for these children? You said that there was no reversal. Do you think that really getting to understand this interface between the microbiome, the glycocalyx, and the neurotoxicity, do you think in the future there is possibly something that could lead the way to an amelioration of the effects, maybe not a complete cure?
Yeah, so the real answer is that the cure is very simple, stop eating cassava, except you can't stop eating cassava because that's your only food source. So really this is a disease of poverty. If you have money, you will not have Konzo. And I don't know many diseases that are a disease of money. This could have been the microbiome and poverty, and this is a disease of poverty. So poverty fixes Konzo. You don't need any science or medicine to fix Konzo. You get rid of Konzo by providing a food surplus in the region so that they have time to process their cassava correctly. If no one's starving, no one's going to get Konzo. It's very simple. But what is interesting of this research is one is that it's very understudied. It's not the hottest type. It's not Ebola or HIV or polio. All the favorites in Congo. But it's a very important topic because it's a food source, a very important food source that is capable of affecting, one's mobility and obviously, destroys economies and villages and one's health. But as global climate change is on the horizon, is happening, food sources are going to become different, and more people are likely to rely on cassava as a food source. Just with the Ukraine war, we see, the estimates of food scarcity and food shortage are out of the roof because Ukraine and Russia produce, 40%, I think, of the world's wheat. And without such food, people that are already in poverty have no choice but to eat cassava. And if you don't know what you're doing eating cassava, well, you're going to have Konzo a lot. So the importance there is really the more research you do on such a topic, the more interest people have. I can't propose, say, to the Gates Foundation, with $2 million, I bet we can eradicate Konzo at least in these villages because we can build a surplus of food. I think you can cure the entire disease this way. But to understand if there's a reversal or actually what's happening, because we have no idea what the mechanisms are. In the lab now, I'm working on making animal models so that we can test, for all I know, this disease is reversible. I have no idea. We've never tried. No animal model exists to test such a thing. And because we don't know exactly the site of damage in the brain of these children, we don't even know where to target, because they have done an MRI or two a long time ago. But nothing, it looked completely normal. But, like diseases such as ALS, on an MRI, you also look normal, even though your motor neurons are deteriorating. You can't detect such things on MRI. So with an animal model, we'll be able to actually tell where the site of injury is. And if the site of injury is not cellular death, but rather perhaps cellular immobilization, maybe they've stalled the cells in such a way, you could potentially reverse it, which is the ultimate goal. And this is where the research is leading. But on the microbial front, no one knew why there was such variability. And this may not, the microbiome doesn't cause Konzo, but it is one of the factors that are, it's a modulator. And I think this extends far beyond cassava. It can extend to all foods. When you're on a monotonous diet of anything, who knows what's happening? Because lactic acid bacteria are great for the gut microbiome. They're very healthy. Everyone tells you to eat yogurt, and yogurt is nothing but lactic acid bacteria, which is 99% always safe and great for you. Except if you were to have a lot of lactic acid bacteria and linamarin, because now you have a scenario that can produce cyanide, which most of the world doesn't have to worry about. But, this is just a chunk, but maybe there's a whole bunch of other kind of scenarios where food source and the gut result in a very unfavorable effect.
I haven't seen any other questions coming up yet. So no hands. So I'm going to keep asking the questions that I have in my mind. When you looked at the microbiome, I've always been of the understanding that the microbiome includes various bacteria as well as fungal and viral elements. Is it possible to look at more than bacteria? Did you focus only on certain bacteria?
Yeah, great question. Yes, in the data set, there is indeed fungal and viral and archaea. However, the gut microbiome as a whole is a very novel, we don't know the bacteria that are in there. The vast majority of these aren't growable in the lab. So we don't know what's in there. So we have to take time to assemble genomes and hope we are identifying new bacteria. The same is true for the viruses. The human virome is even more understudied than the gut microbiome. But to really study it, you have to sequence extremely deep. So these children, we got 5 million DNA sequences per person, which at the time is good. But we've done a follow-up recently, and maybe in a year or so we can have another presentation on what's new. But instead of 5 million, we did 50 million reads, and 50 million reads, 50 million DNA sequences per individual will be able for us to assemble novel genomes of both virus and bacterial and fungal. Because I didn't have the time, but yeast, for example, Yeast is a very important component in fermentation. And yeast also has a very, very high ability to lyse lanamarin. So it's possible that some of these children's guts have a yeast imbalance, if you will, which we are actively currently looking on just for that reason. So yeah, there's many different angles we could take, except once we go below bacteria to virus, to archaea, to fungal, or above or below, however you want to go, our knowledge becomes smaller and smaller and smaller. So it's hard for us to really test a hypothesis with such unknown space. But yes, that's what we're doing, is the answer, short answer.
We have a question. I don't know if you can see the question as well. Hermie from the University of the Philippines. If a less toxic form of cyanide like thiocyanate is present because there are microorganisms that can metabolize and convert lanamarins to a less toxic form, does this mean that there's a less probability of getting Konzo? Can you see that question?
I'm reading. Good question, Hermes. And I'll be in Philippines in a few months to actually start working on some of this as well. If a less toxic form of cyanide like thiocyanate, so thiocyanate is the thiocyanate is a detoxified form. I don't know many plant species that have thiocyanate as a whole. So in theory, if that were the case, thiocyanate would indeed be less toxic, but I don't know a scenario where that exists. However, since you brought up thiocyanate, what is important to note is that while I focus on lanamarin producing hydrogen cyanide, when it's detoxified, thiocyanate itself is also toxic. So for now, we have no idea whether if you're ingesting a lot of lanamarin, then you're also producing a lot of thiocyanate. And it could very well be this excessive thiocyanate production that is causing some of these symptoms. It may not be direct lanamarin cyanide kind of, it could all be thiocyanate, which has been speculated. It's hard to prove, but certainly reasonable. So I'm not sure I'm answering the question, but generally speaking, if you're eating something less cyanogenic, then certainly you will probably have a less likelihood of contracting or of developing Konzo, is a short kind of answer.
Thank you. And Kosinati, you had your hand up. Would you like to unmute and ask a question?
Hello, can you hear me?
Yeah, we can hear you clearly.
Oh, thank you so much. Thank you so much also for the presentation. It was very interesting. So I just have a few couple of questions. Number one, I want to find out maybe the method of isolation that you used in isolating your microorganisms, because I'm also doing the study which is almost similar to the one which you are presenting. So the method of isolation, and also when it comes to your analysis, maybe the tools that you have used that you can also advise maybe somebody else to use, which you found to be more effective. And also maybe the number of samples that you collected during the study, maybe to give you a significant number or to give you like a quite reasonable number that you can work with. And then lastly, I don't know if it's possible to maybe have the presentation, because I would like to go through it again and just maybe learn more of some things. Thank you so much.
Yeah, good questions. So like I said, if you didn't jump on in time, the paper is the same title, The Gut Microbiome in Konzo, and freely accessible. So all of the data, the analysis pipeline, and the actual code to do such is all freely available. So everything we did from computational to isolation is all in public space domain, freely downloadable. If you are in the bioinformatics space, you'll be able to understand the coding for sure. Sample prep is a great question, because this is where I think most people fall very short in the microbiome space. What we did, we go out there ourselves into the field, and we have plenty of liquid nitrogen, and we flash freeze these stool samples. So we try from human to nitrogen. I like no more than 10 minutes. You know, bacterial division is every 20 minutes. So you can imagine if you leave stool setting out too long in the hot sun of Congo at about 37 degrees, you're going to start you're going to imbalance whatever system that you're in. So if you have a system where you do not freeze them readily, then you should expect that your data downstream is not going to reflect the true microbiome because they haven't been processed correctly. And there are plenty of papers on processing. So you could always email me after and I can send you the exact, what I would recommend. Secondly, for the DNA isolation, this is also where many people can go wrong. Bacteria are, there's gram-positive and gram-negative. So some have cell walls, some don't. So in the isolation process, you got to make sure that there's some kind of mechanical disruptor. Bead bashing is what they call. So there's a whole bunch of beads that rip apart the bacteria so that you can really get all the different types of DNA out. And in cohort size for this paper, we were able to do 30 per group, and it was significant only if the effect size is big, of course. In our follow-up, we've done groups of 100 instead of 30, but it comes at a very, very high cost, to do a follow-up of an N of 300 at 50 million reads. So we did metagenomic, metatranscriptomic, metabolomic, and we're talking in the ballpark of about $250,000 US to actually do this at the depth and quality that it is required. Of course, you can always do different levels of, experimentation and finances, but that's my recommendation. But again, like I said, that comes at a cost of 250,000, and which is not available to most. I got lucky. Most people in the US or anywhere don't have those kind of funds to do such a thing. But yeah, in the paper, check the paper out and email me if you have questions. But those details are very well described, and free bioinformatics code on there as well.
And also this presentation will be available on YouTube on the UKZN channel. So you'll be able to see it by, I would guess, the Easter weekend, by next week at least. Are there any other questions? Also, just to let you know that Matthew is with us until the end of next week. If anybody would like to set up a face-to-face meeting to talk to him. Are there any other questions before I ask another one? Okay, while we're waiting for other people to ask, Matt, you and I are about to embark with a student who's got a research question on looking at the oral microbiome. Would you like to expand on why looking at the oral microbiome might lead us to some conclusions about health?
Yeah, so oral microbiome, even less is known on that than the gut microbiome. So in the space, there's a recent paper from a collaborator of mine, Ren Blackman, also freely accessible, and he did a review on the literature essentially that tackles microbiome. And very much like human genetics, it is predominantly focused at the United States, European, Western countries. Sub-Saharan Africa is very unknown. It's an unknown space from all microbiome. Southeast Asia is a very unknown space of all microbiome. What's important about understanding different orifices of microbiome is, your mouth is your first line of everything. And say, for example, you have, just think of a disease state, you have periodontal disease. That doesn't just affect your teeth. You're swallowing all those bacteria and all those toxins. And generally, it's always well known that oral health can be reflected in other types of health. Having an abscessed teeth, it doesn't just affect your tooth, it can kill you, right, if the bacteria spreads. And interestingly, in the oral space, I can actually bring it back to Konzo, believe it or not, because unprocessed linamarin is actually quite prevalent in tears and saliva. So you could also see a scenario where understanding the oral microbiome, because imagine if my saliva is also rich now in linamarin, and I have bacteria in my mouth that is producing hydrogen cyanide from that linamarin at this level, as opposed to inside, who knows what that does? And the topic is on different oral and throat cancers. So you can, imagine if you have a dysbiosis in your mouth, perhaps there's a bacteria that's producing some, unfavorable toxin leaching down your throat 7-10 years. You might get some type of cancers, and if you smoke or if you don't brush or if you, different alcohols, who knows? But all things could have an effect, but we don't know. It's a very unknown space. Mm-hmm. Maybe in years we'll know better. I mean, we understand how bacteria in the mouth affect dental health, but that's not what we're talking about here. That's well established. How it affects global health. And you can imagine it probably affects your gut bacteria as well, because you're swallowing this 24/7. So whatever, if you have a dysbiosis in your mouth, you could probably lead to a dysbiosis in your gut just as easy, just from an unhealthy orifice here affecting another area of the body downstream. And so it's a very important thing to know, just a very unknown space, particularly on this continent.
I think talking about dysbiosis is important now, because people don't realize that our microbiome has evolved with us. We've got a healthy microbiome that helps us survive in this world. And dysbiosis means when that goes out of kilter. And
Exactly.
So often, and this worries me, and I might be speaking out of turn here, but when you go to the doctor and you get an antibiotic, they give you a single probiotic. And sometimes I wonder, do we really understand? Is just giving a probiotic, and some of them might have 4 or 5 strains in them, is that really helping? Especially if you take an antibiotic every day for 5 days.
Exactly.
Is it helping? We've really got to get to know what we're doing with causing dysbiosis, because trying to recover from dysbiosis, first of all, we don't really know how to do it. People are doing fecal transplants.
Yeah.
But this is a new area. It's a new science. Yeah. Are there any more questions? Sorry, carry on your comment.
And that's why it's exciting. Yeah. We don't understand, we know a dysbiosis. We have no idea how to fix that. Probiotics, plenty of studies have shown it doesn't work, because if you think about it again, like an ecosystem, you can't just throw any random seed into a forest. It has to be placed correctly. If you put a seed under a canopy of trees with no sunlight, that thing's not going to grow. It's a very complex kind of arrangement of where bacteria live and who they talk to and who their neighbors are. And they just don't let anybody in, You just can't take a bacteria and it all of a sudden becomes a colony in your stomach. You probably just, defecate it out, and it never becomes, it's not part of you. It's just in your digestive system.
Yeah.
So
I was also interested in right in the beginning of the talk, you spoke about sulfur. But if a kid had a steak the night before, he wouldn't get Konzo, because that's particularly, I find that particularly interesting, how sulfur keeps the glycocalyx healthier. If you've got an undersulfated glycocalyx, you've got problems. So I remember years ago in the 80s when I was studying, there was some campaign that said if a child ate 1 egg a day, they would never be malnourished. And I can actually understand that now. 1 egg, it's so nourishing. But how do these little children who can't get anything but cassava get eggs?
I know. And this is the thing. It's a vicious circle. Poverty leads to a food shortage, and then you become hungry, and then you become rushed. So you have to produce, if you have 5 children and the mom's working in the field from sun up to sundown, there's no time to prepare things. Yeah. But again, we've thought about, I've thought about the introduction of a large, you got to introduce it so that it's overwhelming. Everybody needs 10 chickens, because if somebody's producing eggs and you're broke, then you're going to sell those eggs to make money. And it makes sense. If you're poor and you have eggs, you sell the eggs. Yeah. But if everybody has an egg supply, then nobody needs eggs. So therefore you can eat eggs. And so those are, reasonable kind of potential solutions for certain diseases like this. And it doesn't take $1 million of science. It could literally just take chickens, or something. Who knows? But a chicken egg would solve this problem as well.
Yeah. Good. I think it just leaves me to say thank you very much, Matt. Would you mind putting your email address in the chat box? We cannot send people your papers, but if you've got your email in the chat box, they can approach you personally so that you can link them to your ResearchGate profile.
It's just mbramble.ucla.edu, so easy to remember.
Do you have a ResearchGate profile that they can look up your paper?
Yeah, you can also.
So just go. Look for Matthew Bramble on ResearchGate, and you'll find it all there. Yeah, I love ResearchGate. You don't even have to.
Me too. Most of my work is open access anyway, so I don't like publishing in a non-open access.
Yeah.
Unless I have to.
I'm looking forward to collaborating with you on a different type of study now. Still genetics, but completely different. Thank you, Matthew, and have a wonderful weekend. And thank you for everybody who attended today. Thank you for taking the time while everybody has already started their holidays. and we'll see you again soon. Bye-bye
Take care.
then.
Bye-bye.
Bye-bye.